All right, people. Happy Friday. I am missing Amra, Claire, Olivia, Vance Thomas. If any of those people are here, say something. It's magic. Bilk? Where are you getting bilk from? I think you guys need to check your spelling. Anyway, oh, I got a great idea. Remember when I told you to learn schmooze on your own? And everybody's like, oh yeah, we're going to learn schmooze on our own. Like, sure thing, Bulk, you can trust us. We totally always do what you say. We totally always work on the stuff you give us to work on. We don't ever blow it off and not do it. Anyway, I'm thinking we should have like a little mini quiz, a fun quiz on signal transduction to see if people learned the calcium pathway, G proteins, receptor tyrosine kinases, you know, that stuff you're supposed to memorize. Let's have a little mini quiz, 4 questions. I'm not going to say it's easy, but if it was easy, it wouldn't be fun. See, it's a fun quiz, means it's challenging. Yeah, it's formative. It's not going to count. Don't stress. It's Friday. It's more like a fun game, you know, like prove to me what you know kind of thing. So don't stress. Not going to hurt your grade. It might hurt your pride, though. That it might hurt. Your self-esteem may drop just a little, which I think is good for some of y'all who've got this escalated, elevated self-esteem. So, Findeligs, yeah, that's great spelling. Hope you can spell better on the quiz. All right, let's take a look. Let's shall. Oh, look at that. There's already a quiz there just waiting for us. Where did that come from? My goodness. I'm going to give you 5 minutes from 30 seconds from now because it'll take a minute for you for it to pop up for you guys. Only 4 questions, multiple guess, 5 minutes. I'll set a timer. And yes, you can use notes if you must. It's just for fun, so it's not like it's cheating. But the way I phrased the questions, I don't think it'll be all that easy to use the notes. You got to kind of know stuff. We'll see. We'll see. All right, easy with the chat now, and let's begin the quiz, okay?
Volk, I can't find it.
It should be under Unit 2, Cells, Signal Transduction Quiz. I'll move it to the very top if that helps. Signal Transduction Quiz. Refresh your browser if you must. If anybody else is having trouble seeing it, please let me know. Please yell it in the mic. Is everybody good now?
Oh.
Yeah, what's up?
For like the like where it says select all that apply, it doesn't work in like Google Forms.
Can you say it louder?
Only let us click one of the select.
Yeah, you can only pick one.
Oh yeah, I noticed that. That's actually I didn't realize that's kind of like a little flaw in Google Docs that only one will work. So there's 2 right answers there. If you pick one of the right ones, you'll get the point.
Okay.
Does that make sense? So you got a 2 out of 5 chance of getting it right. All right, I put the timer up on the screen.
Mr. Volk, you're muted.
Was I muted this whole time I was talking?
Yes.
Yes,
Yeah.
you were.
All right. Well, that's good because I was saying really stupid stuff and I kept saying I pronounced apoptosis wrong. So I'm glad it was off because you never heard me say it wrong. So it doesn't count as a mistake.
You're still recording though.
Yeah, but you just admitted to the mistake.
Ooh, I'm gonna have to edit that out. No, it doesn't record if I have mute. It won't record it. It only records if I don't have mute. So I'm good. I'm good. I'm good. I don't have to edit it. Yeah. Yeah. Missed it. No. Oh, no. You don't know if I talked because you didn't hear me and you didn't see my face because I was aimed at the ceiling like this. So therefore, no evidence. Okay. You exceed. We heard you. Yes. Yeah. Okay. I guess that's a mistake on you. Exceed. Claire is not popping in as usual. Man, I hope she gets her internet fixed. My goodness. Okay, first of all, if you take a look at this screen here, let me just mess with something here. Hold on a second. Screen 2, this is screen 2. Yeah, if you take a look at this for a second, this is usually I have kids make a poster. And obviously this year we're not gonna like go in groups and make a poster. So what we're going to do instead make a Jamboard. We're not going to do that in class. I'm going to like just throw you in a group randomly sort of, and in your group, you're going to make a Jamboard that covers all of this stuff. You can forget about this. You can like leave this out. Oops, do I get to draw on this or not? Yeah. This you can leave, oh no, yeah. Okay, this you can leave out and this you can leave out. Okay, but schmooze and apoptosis you can throw in there. And then the main thing of the poster or, Jamboard or whatever is basically an overview and summary of all of signal transduction that we talked about. So if we take a look at that, basically we start with a ligand and let me just see if I can do my pen thingy. I'm in Microsoft Paint, so it's not ideal for the pen, but might be okay. Okay, so we start with a ligand, and then there's two options. The ligand can either be water soluble or it can be lipid soluble. So if it likes lipids, if it's hydrophobic, it'll go through the membrane and it's like a steroid or something like that. And that's these guys here. And we've spent very little time talking about these guys. So this would have an internal receptor like a steroid, which then activates a transcription factor and then, makes protein synthesis happen. All right, so that's one option, and that's just kind of a small part of it. But then the main part we talked about in chapter 11 is what if the ligand binds a membrane receptor on the outside of the cell? What about that? And so we said there's three main options. In real life, there's more options. It's more complicated, but for our purposes, we simplified it and we said these are the three main ones, and these are the most common. G protein coupled receptor, activating the G protein pathways. And there's two of them that we talked about. RTK is another one, so receptor tyrosine kinase, whatever it does. And then, this we could almost do like spreading out because it's, broad response. Hey, did you catch that on the quiz? That was one of the questions. Which one has a broad response? Everybody caught that, right? Everybody knew it was RTK? All right. What is LGIC? Anybody know? I'll give you a clue. Ligand.
Channel gate.
Ligand gated. And Max, you're right, but let's actually spell out the acronym. Ligand gated.
Ion channel.
Ion channel. Very nice. Okay, so that one also we did not talk about very much. There's not much to say. It opens the channel, the ions rush through down the concentration gradient. That's about it. So really most of the time we spent on, these guys here. We spent a lot of time on those. So I'm going to ask you and your group to make a Jamboard, and I'm going to ask you to make beautiful pictures as if you were making a poster on paper with markers and, colored pencils and stuff like that. Make a beautiful picture of each of these mechanisms, and I think that's going to help you learn them better. Because you're going to have to think about like, hey, how do we organize this? What happens next? What is this thing over here? What's released here? Blah, blah, blah. So just to make sure that this is actually possible, like can Jamboard actually do this for you? Like, is it even like a good idea to use it? I spent about, I don't know, 10, 20 minutes this morning, and I just tested it out. And I just thought, hey, let me try to do the most complicated one of them all, which is calcium, phospholipase C. That's the most complex one with all the most steps and all that stuff. Let me see if I can pull that off on the Jamboard, because if I can do it, then they can do it. And if I can't do it, then, it's going to be pretty hard for them, and it's not really a fair assignment. So let's take a look. Where am I going? Timer. Oh, I don't need the timer anymore. Okay, so let me show you what I came up with. And I'm not saying like copy my thing here. I'm not gonna like even give this to you. I'm just gonna show it to you. It gives you an idea of what's possible, and that it's definitely doable. And it didn't take me super long to do this, especially once you figure out the little tricks of the Jamboard tools. And it is very primitive. Like there's a lot of things you cannot do. I thought it would be a great time to tell you this class is causing me pain. Well, I've shared with you what this class does to me, so you know that. But check this out, like pretty nice, hey? Like I made that, I'm kind of proud of it. It looks decent. I use the text thing to just label my little parts. So my G protein coupled receptor over here, it's right here. I got my ligand binding, I got the G protein with GTP attached, then goes over to phospholipase C. I made PIP2 kind of primitive. I just made like the DAG into a square or a rectangle, and then the IP3 into a triangle because it has the 3, phosphates stuck to it. So that's my PIP2 that gets split up into DAG and IP3. This goes over to, I didn't label the ion channel. So you still have to label the ion channel, or I would have to if I was going to hand this in, and then channel opens up, calcium is released, calcium binds protein kinase C, which then elicits the cellular response over here. So, I would say this is pretty decent, not bad. And it could actually help you learn the process better by doing this. So this would be one of the, slides or pages or sheets of the Jamboard. And then over here, you could do adenylyl cyclase. So you start again with G protein coupled receptor, draw the membrane in the way I did, and just do your best with it. You can freehand it if you'd like, or you could use the shapes. The shapes are pretty easy to use. So let's say you want to make like adenylyl cyclase. I would just go here, make like a circle or something. I'd say, okay, here, this is my adenylyl cyclase. Make it like whatever you want it to look like. I don't care. The shape doesn't have to be realistic or anything like that. And then cyclic AMP, you could, since it's cyclic AMP, I guess we should, make like this is like your cyclic AMP, then make it a certain color by just coloring it in like this, whatever. So lots of options. And then I guess basically what I'd like you to do is in your group, you assign amongst yourselves, you assign who's going to do what. So in your group, you guys are going to decide, get together like outside of class sometime over the next, week or so, and meet each other in Zoom. And then, or you can just text each other or, just email each other or whatever, and say, okay, I want you to, I volunteer to do the calcium one. I'll do receptor tyrosine kinase. I'll do cyclic AMP or whatever. What else is on the thing? Let's see. Let me get rid of this here. Hold on. Wait, can I undo the, wait, what? Oh, why did it do that? I don't know. Okay. Oh, the ion channels is, whoever does like RTKs, because that's kind of much smaller, they could also do the ion channels, or maybe the person that does the ion channels also does the internal receptor. Let's say it's 4 people in the group. So then it's not so, unfair or whatever. Can you pick your groups? Well, sure, but I mean, sure. Yeah. How about this? How about this? I'll just leave it completely up to you to, if you want to be in a group, pick a group. If you want to do it by yourself, then you have to do all of it. If you want to be in a group, then you guys can divvy it up and hand it in as a group. How about that? Deal? So then I don't have to spend class time right now assigning groups and people like, I can't work with them and nobody wants to be with Noah and like all this kind of stuff, whatever. But you know how that goes. Anyway, and if anybody if somebody's shy and doesn't want to like ask people to be in the group or whatever, just talk to me about it and I'll find a group for you. I'll I'll find some people that you can work with, and it's fine. We'll work it out. We'll figure it out. I wouldn't admit that in public if I were you. Anyway, yeah. Okay, so that's that. And this this poster guideline is on the Google Classroom. Let's check that out. I already put it here. It's called Signal Transduction Jamboard. I gave you really no instructions because I just whipped this together real fast this morning, and I didn't have a lot of time to like give you instructions. So it just shows the picture, that's all. But it's, you use Jamboard to do it. And if you have another vehicle, like other than Jamboard, that you want to use, if you want to use some other program, and fine, you know, whatever. If you want to do it on paper and then take pictures of it, you know, that's okay. But the Jamboard thing worked all right. I thought it was pretty easy to use, and it wasn't too bad. How do we do on the quiz here? Summary, the average was 1.77. Ouch. Ouch. Oh my. Oh, this is the scores. We can't look at that. Question, let's see. Yeah, all right. Anyway, so it looks like some of y'all didn't study your schmooze, huh? Okay, let me get rid of this here. Just delete that real quick because that is, oh no, that's my good one. I need to get rid of this. Right, am I sharing? Yes, I am. Let's go to PowerPoint and let's talk about apoptosis. So I talked to you guys about apoptosis has like, there's two major functions of it. One is development, okay? Like getting rid of the tissue between the fingers in the embryo so that the fingers can form separately, and lots of examples like that. Lots of parts of the body form where more cells than you need will form up front during development, and then some of the cells die back, and you know the other ones will continue to form the final body shape. And then the other function was to get rid of cells that have gone rogue. So sometimes cells get defective and they got bad genes, too many mutations, the DNA is damaged maybe or something like that, or they become cancerous, or you know other kinds of damage. Then apoptosis gets rid of those cells, it destroys them, and white blood cells will eat up the blebs, the fragments, and you know sort of recycle those materials, which is a nice system to take care of the body and keep the body healthy. So pretty good. So apoptosis overall, even though it says cell suicide or cell death, sounds really bad, it's actually a good thing. However, if apoptosis goes out of control, it can also cause problems. And some of the neurological diseases like Parkinson's and Alzheimer's and stuff, they think it might have to do with apoptosis. We don't fully understand those yet. All right, let's take a look at the mechanism. So this was worked out in an organism called C. elegans. And C. elegans is a roundworm. It's a primitive creature, but it's widely used in research, in developmental research, neurological research, a whole bunch of different areas, genetics, all this stuff. So it's a well-studied organism. And this was where apoptosis was first worked out. So our mechanisms in the human for apoptosis are very similar, but they're not exactly like this. This is a nice model to help us understand how it works in the human also. All right, so we're only going to worry about the C. elegans version of it because it's simpler and it's easier to understand, and it's also the one that's best understood by scientists. Okay, this is a little bit scary because the deal with apoptosis is the way that it's set up and the way it's regulated seems a little bit backwards. It seems very risky and maybe not wise to set it up like this. But this is what nature has come up with, or you know, evolution or God or whatever you want to attribute it to. This is what we have, and this is what we have to deal with. Basically, what we have is we have two caspase enzymes, SED4 and SED3. And by the way, SED stands for cell death, which also sounds scary. SED4 and SED3, or at least SED4, is naturally active. It wants to kill the cell in its natural resting state, and it will proceed to kill the cell and activate, the blebbing mechanisms and the breakdown of the proteins and nucleic acids and all of that stuff in the normal state. Like it always wants to do that, which is kind of crazy. And the reason it doesn't in most of your cells all the time is because SED9 stops it. So SED9 blocks this. It inhibits it. It shuts it down. SED9 prevents it from killing the cell under normal circumstances. When SED9 gets inactivated, so if this SED9 protects you, it protects the cell. When SED9 gets turned off for some reason, by some mechanism, now SED4 spontaneously becomes active because that's its natural state. And apoptosis will proceed. It will activate SED3, and then a variety of mechanisms are going to go into effect that then will cause blebbing and cell breakdown and cell destruction, cell death. Ironically, the SED9 is stuck to the mitochondrion, and that isn't really all that significant, but it can be stuck to mitochondria, and it has really nothing to do with the rest of the function of mitochondria. It's just a place where they sometimes are kept and stored. My analogy for this is the following. It's a very odd analogy, and please don't, get upset or whatever, but I know you're not really supposed to be talking about guns and stuff like that in school or whatever, but I'm going to talk about guns. Like a handgun is my analogy, and here's how it works. Normally on a handgun, we have like a safety, we have a trigger you have to pull before it shoots and can actually hurt someone. This would be like a handgun that if you left it alone, if you didn't touch it, it would be shooting all the time. If you just put it down on the counter, it's shooting bullets. And in order to stop it from shooting, you have to push a button or you have to squeeze the trigger, and that'll stop it. And as soon as you let go, it'll shoot. That's what SED4 is like. Now that to me is scary as heck. That to me is freaky. Like if I have to do something to get it to stop doing the horrible thing, and if I stop doing it, if I forget to do it or whatever, it'll start shooting, which is weird and bizarre, but that's how it is set up. Everybody clear on this? Okay, if you're offended by my handgun analogy, I apologize, but anyway, it's just the best thing I could think of. Because I think it's a good analogy. It's just whatever, controversial. All right. Well, what might cause SED9 to become inactive? Well, if there's a lot of cell damage, there's mechanisms that inactivate SED9, and then the cell automatically breaks down and gets destroyed. It breaks itself down. It literally is cell suicide. Another mechanism would be there's actually a receptor on the outside of some cells that when a certain death ligand binds, a death ligand will bind the receptor, it will trigger SED9 to become inactive, or it'll shut it off and then activate apoptosis to kill the cell and then destroy the entire cell. So you might wonder, like, why in the world would a cell want to have a receptor that causes its own death? Like if I was a cell, I wouldn't, like, I don't want to have a button on the outside of my body that if somebody pushes it, I explode. I don't want that. Or like I disintegrate or burst into flames. I don't want that. Like that button seems like a bad idea to have. Why would a cell want that? We already know the answer, so you should know the answer already. Let's see what you come up with. I activated the chat so you can type again. Or you can say it. When would a cell want to get destroyed? Or when is it a good thing in the body when a cell gets destroyed? Other than like damaged cells.
When it keeps on multiplying.
Can I say it louder?
When it keeps on multiplying.
Okay, that's actually like if a cell has gone rogue and bad, and then it gets destroyed by apoptosis to make sure that it doesn't form cancer and stuff. I'm talking about another reason where there's actually a signal from the outside that tells it to stop.
It's like infected with something.
That also would be the other possibility.
If it presents
But okay, the whole Max, the whole infected thing, that actually involves the immune system a lot more. There's a lot more complicated mechanisms that'll break those down, and we may talk about those second semester. So that's actually a whole nother thing. That's not just straight up apoptosis. That it involves the active immune response. Okay, somebody else was saying something and I interrupted. I apologize. Go ahead now.
Like if the cell prevents a danger to the organism.
Nope, that's also that other one. All the things you guys are guessing are when a cell goes bad and presents a danger. Think about tiny little baby you. think about egg and
the
sperm.
hands.
Zygote,
Hands.
developing. Ah, hands. Well, it's not just hands, but hands is a good example. It's a good idea for these cells to die so that the hands can form in the early embryo. Are you with me? So the fingers can form correctly. So those cells have a receptor on them that allows them to break down, that allows them to engage apoptosis. I almost said it wrong again. Oops. All right, so that's what I'm talking about. So these would be like the cells that then have to get eliminated during development. Those are the extra cells that were formed during development that then have to be destroyed. All right, so anyway, keep in mind those cells don't have hopes and dreams. Those cells don't have a mind. They're not sentient. It's okay for them to get removed. It's all right. It's like if you build something out of Legos and you remove some Legos to build a window in your little house that you build, and that you don't have to worry about hurting the feelings of those Legos. That's basically what this is like, okay? Before you start forming protests outside of school, you know, because I'm killing cells or whatever. Anywho. All right, so let me continue. So caspases are the proteases that take care of this. Caspases are the enzymes of apoptosis. Okay, that's what they're called. Apoptosis can be triggered by an extracellular death signaling ligand, like in development, when you have to remove certain cells. DNA damage in the nucleus, those cells are going to go rogue and they might become cancerous, and that would be a good idea to destroy them. Protein misfolding in the ER, if the protein is folding incorrectly, the cell might present a danger to the rest of the body, and that cell needs to be destroyed, and that might trigger apoptosis. So a bunch of these different kinds of, when things go horribly wrong, events may trigger it. All right, what else do we have? Oh, yeah, I think we're almost done here. It evolved early, mostly for development and maintenance. So that's why because it evolved early, that's why mechanisms of apoptosis are real similar in humans and in C. elegans, like the worm. Because the chat makes me want to stab my eyes out, seriously. Like I might just wash them in bleach and just be done with it, because like reading the stuff you guys type is just, it's too much. It's too much. So because it evolved early, that's why the mechanism is similar. Okay, that's kind of like evidence for evolution. So that's kind of neat. Apoptosis may be involved in stuff like Parkinson's and Alzheimer's, where brain cells die prematurely, where brain cells aren't supposed to be getting killed. And that's when something has gone wrong with apoptosis, when it's not getting shut off. Like something's wrong with SED9 maybe, and it's not turning off apoptosis, and you have too much of it, so then cells get destroyed. So that'd be like the opposite of cancer. Cancer is too much cell growth, too little apoptosis. Parkinson's and Alzheimer's and other degenerative diseases where cells are dying too soon, we have too much apoptosis and not enough cell, healthy cells and stuff. All right, here's that hand thing that I talked about. Okay, that's just a little fluorescent microscopy here. Shows it very nicely which cells have to go away, and they're actually glowing like yellow right here in the middle. Those are the cells that are going to have to disappear. And then there's some questions at the end that are good review questions, and you can check those out on your own. I'm not going to hold you to those or check those or anything like that, but that's extra stuff you can do if you're ambitious and you have too much time on your hands. That's something that you can look into. Okay, any questions on chapter 11 on signal transduction, whether it's G proteins, whether it's RTKs, whether it's apoptosis, whether it's schmoo? Any good questions on schmooze? I wasn't going to go over them, but you're supposed to learn them on your own, but guys get the schmooze. Did you know that it was, so what are the two letters for the two mating factors? Is it A and B? Is it alpha and beta? What is it?
Alpha and A.
Alpha.
It's A and alpha. Isn't that weird? A and alpha. It's like you don't expect, you expect alpha and beta or A and B or something like that. But I'm going to have to check later how many people actually got that right. Okay, cool. What I'd like to do next then is I would like to go into chapter 12, which is mitosis. And mitosis is going to be a lot of review. The first half or first third will be review. And then the second part will be new stuff that's going to be a little bit hard, but not as hard as chapter 11, not as complicated. So you should rest easy and be confident that chapter 12 will not be as bad, but it's not going to be child's play either. All right, so we'll do, for the first third then, or the first part, I'm going to go pretty fast. For the second part, I'm going to go a little bit slower. So before I forget to announce this again, even though I announced it last time, I know that you guys forget things. Maybe I'll also put it in the stream in the Google Classroom, if I remember. I'm planning on having a test on a week from Monday, or not Monday, when do we meet? Tuesday. Yeah, we are on Tuesday, right? Yeah. So a week from Tuesday, I'm planning on having a test on chapters 6, 7, 11, and 12. I'm going to get through hopefully more than half of chapter 12 today. I'm going to finish chapter 12 on Monday, no, Tuesday, when we come back. Then it's Thanksgiving, so we don't have class. If I don't get done, I'll give you videos to watch over Thanksgiving, and I'm going to give you work to do over Thanksgiving, just so you know. You're going to get work. So I understand you want to eat turkey, and oh yeah, that's fine. You can do that on Thanksgiving, but you do have Wednesday. You have Wednesday off. You don't have school. Spend time with your family on Thursday, eat your turkey and all that stuff. Friday, do your Black Friday shopping early or do it online. I just do all my Best Buy shopping online and get all the deals and don't have to go out. And then you have time Friday, Saturday, Sunday to work. I've already posted in Google Classroom free response questions for you. So let's take a look at those real quick before I forget. What happened? Why did I close it? I closed it? I was dumb. Okay, so I think I posted these already. I hope I did. Oh yeah, I did. So I got my Jamboard instructions here. We just did the quiz, and then we have these free responses. Unit 2 cells, review FRQs. All right, you click on this and you can make a copy for yourself. Now this is going to be a little bit intimidating at first because if you look at how many free response questions there are plenty. There are a lot. The good deal is, and the last 3 are like actual AP questions, and there's some stuff in there that we haven't learned yet, like prokaryotes over here. We haven't learned that yet, so you don't have to worry about that part. And I told you that. Somewhere it says, note, for the previous question, we have not covered part B. Attempt an answer, like come up with something, but you don't have to worry. I'm not going to ask you that on the test. But the other stuff is pretty straightforward and stuff that we did. Here's apoptosis, C. elegans, la la la. So water potential, organelles, all that stuff. The question is, do you have to do it and hand it in? And the answer is, right now I'm leaning towards no. I'm giving this for you to practice for the test because you know there will be questions on this, from this on the test. I will slightly modify them and I will put them on the test similar to the way these are. And there are certain ones that I like the best that I'm going to put on. You know damn well I'm going to give you water potential. You know I'm going to put one on there because you know I like that. There's going to be adenylyl cyclase or phospholipase C, guaranteed, or RTKs. I really like that stuff. So, anyway. And then there'll be multiple guests. But so that we have in a week from Tuesday. Cyber Monday, Schmonday, forget about it. Crying out loud. Yeah, so no, not a week after break. It's as soon as you come back after break, we have the test. The first day back, we have it. We're done. Crying out loud. I'm spoiling you. The test should have been next week already. You're spoiled. We're like 2 weeks behind right now where I was last year. So we're going slowly. Crying out loud. Anyway. What did you guys think this class was going to be? You thought like, oh, this is a class where we're going to learn about giraffes or something. That's what you thought this was, huh? Wow. We're going to look at puppies and things. Anyway, so that's there for you. I would recommend that before Sunday, not this Sunday, but next Sunday, you get these done. So that on Monday, you don't have to worry. You got them all done. And then on Tuesday, when the test comes, you're not going to have to sit there and like, anyway. Yeah. I'll make you a deal. I'll let you use all this then on the test. You can use it. You do them, you can use them. Okay? So Should be easy. If you don't do them, you can Google all you want. You can try to cheat all you want. You're not going to be able to do it, which we found out on the last test. Well, some of you guys kind of bombed it, even though it's at home and all that. Well, I can just cheat and I can just get all the right. Yeah, okay, good luck. See how that goes for you. Does apoptosis damage DNA? Yeah, it'll break down mostly proteases. It'll break down proteins, but it can also damage DNA and chop it up, some of those enzymes. And then the whole cell will get just fragmented and made into these little bubbles, which then get eaten by white blood cells. All right, it's virtual. Yeah, everything is fake. Everything's virtual. Nothing is real. Yeah. Anyway, do I really have to read all? Okay, I'm not going to read it. Go away, and let's dive into mitosis. So we're going to talk about cell cycle a little bit, kind of review from freshman year. I'll tell you a few new things, and then we'll zip through mitosis nice and fast, because that's easy. You freaking learned about mitosis in middle school already. Then you learned it again freshman year. Now, I'm not going to like spend a huge amount of time on it. I'm going to kind of assume you have the rough idea of what it's all about. I'll refresh your memory. You should probably study it a little bit so you know what happens at each stage and what the point of it is. But then the new stuff I'll spend a little bit more time on. Okay, let's begin. All righty then. Did Claire ever get in? No, she did not. Well, I hope she watches these videos. Claire, we're talking about you when you're not even here. That's kind of mean. Sorry about that. Okay. All right. So mitosis could have a number of different functions. So in humans, it has basically growth and development to me is like one thing or whatever. I mean, you know, obviously it's different, but and then repair. Those are like the two main functions for us is like when you're a tiny little cell, zygote or whatever, and then you got to grow into, you know, a ball of cells into the embryo, blah, 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 into the adult human. That's mitosis. Mitosis takes care of all that. You got to have mitosis to do that. And we just learned you got to have apoptosis to do it because you got to get rid of the extra stuff, the extra junk. And then secondly, if we get injured, if I cut myself, then I'm going to need mitosis to heal that, to repair that, and fix it. Okay. So in unicellular organisms, mitosis acts as a reproductive mechanism. So it causes reproduction. That's not for us. We don't split in two to reproduce, but like amoebas do. Paramecia, they do. They'll split in half. They do mitosis, and that's a mechanism of reproduction. So that's a third function of mitosis, reproduction, but only in unicellular creatures. So not in animals or plants, let's say. When you come across something new, can you say it's new? Well, none of this is new right now. Yeah, I'll tell you. I'll automatically say it. I'm sure I will. Yeah. I understand your question now. I didn't get it at first. Now I get it. Okay. We should know what the genome is. So genome is all your DNA. So in animals and in larger organisms or more advanced organisms like eukaryotes, genomes are made of chromosomes. Okay, so chromosomes are linear pieces of DNA, long, so large, linear pieces of DNA. In other words, the DNA has a beginning and it has an end. Whereas prokaryotic genomes are loops. So bacteria have a loop of DNA. They have a circle, no beginning, no end, and much smaller. Okay, and obviously humans, we have 23 pairs of chromosomes, which gives us 46 total. So we have 23 different pieces of this DNA. And we have one set of 23 from our mother and one set from our father because we're sexually reproducing organisms, gives us a grand total of 46. But we always have two of each that are similar with the same genes on them because of that fact that we're diploid. And diploid means we have two sets of chromosomes, one from mom, one from dad. All right, so that's something that you want to think about and that you want to make sure you know these terms. You should know what somatic cells are. Those are body cells. So regular normal cells are somatic cells. Gametes are not somatic cells. So that's the other kind. So those are like egg or sperm. Those are sex cells. They're haploid. Okay, so these guys are diploid, 2N. These guys are haploid, N. Chromatin is just the DNA spread out. The DNA is all spread out, and the nucleus, this is normally the case for a cell. A normal cell doing its normal stuff is in interphase, and the DNA is not coiled up into the little X's that we're used to. Like when we think of chromosomes, we always think of these X's like this, and that's not what DNA normally looks like. Only right before mitosis does that happen. Okay, and most cells don't do mitosis all the time. Some cells don't ever do it again in our body. They're permanently done with mitosis. So they're always going to have the DNA spread out like this. And not coiled up. All right, and the proteins that help the DNA wrap, they're called histones. Histones, those are proteins that help the DNA wrap up. And so the chromatin is like those proteins and those histones, and the DNA sort of spread out all over the nucleus. Sister chromatids are just these two. So when you see the chromosomes as an X, like this, okay, that's horrible. Let's try it again. As an X like this, these two are sister chromatids. All right, and they're exact reproductions of each other, they're exact duplicates, and they happen through replication when DNA copied itself right before mitosis during the S stage of the cell cycle, which I'll show you in just a second. All right, centromere is just the thing in the middle where they're connected. That's the centromere, where they're stuck together still. Okay, and then eventually during mitosis, they're gonna get pulled apart, they're gonna get separated, the centromere is gonna break, and then each chromatid is going to move to the new daughter cell. All right, so those are some terms that you should know already. Just did a quick review of them. Here's some fancy pictures of all of that stuff we talked about. Your centromere is right here. Here's each sister chromatid. You may notice how right here, the way that it looks in real life, and even in this drawing right here, they don't look like this. The chromatids are actually like really close together like this, like parallel to each other. And so it's like, is that just like a matter of choice of the artist? Do they get to, can you draw it any which way you want? Well, there's actually a reason why they're so close together. There are proteins that are holding these two chromatids together really, really tightly early on during prophase and into metaphase. And those proteins are called cohesins. Cohesins, like cohesion. They hold the two sister chromatids together and make them stick together. That's new, by the way, Ensign. That is not something I taught you in freshman year. The cohesins thing. Everything else has been old stuff. All right, and those cohesins are going to get split late metaphase, early anaphase in order for the two sister chromatids to come apart. But they help them stay together when they get moved around and jostled around during prophase, when they get lined up towards metaphase to go into the middle, so they don't get damaged and get separated too early. All right, centromere here, sister chromatids, blah, 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 and good to go. Oh, by the way, this is a little bit unrealistic. Like this is showing just one chromatid, and then it's duplicating and forming the sister chromatid where there's two of them. The chromatid wouldn't look like this. It wouldn't be condensed. This would all be spread out when it's duplicating. It would all be spread out all over the nucleus, because in order to replicate DNA, you have to open it up and unzip it, and you can't do that when it's coiled up tightly. So that is actually kind of an unrealistic picture to show like a single sausage chromatid like that. That's not really going to happen. By the time you see them coming up as little sausages like this, they're already duplicated. There's already going to be two. All right. Mitosis is division of the nucleus. Cytokinesis is division of the cytoplasm. Okay, and so that like at the end when the cell splits, we'll talk about that again, that splitting at the end, that's cytokinesis. But the splitting of the nucleus is basically mostly what mitosis is about. So this would be mitosis, and then this is meiosis. We're not really talking about meiosis now, though. This chapter doesn't really deal with it, but we should remember what it is. How are they different? So mitosis, we said, is for growth, it's for repair, and it's for reproducing single-celled creatures. What's meiosis for? What's the point of meiosis? It only has one function. It's to make what?
Duplicate.
Yeah, reproduction, but specifically what cells is it making? Nobody's answering me? I'm so sad.
Gametes.
Okay, so gametes, so ova, which are eggs, and sperm. All right, so that's what meiosis is for. So in meiosis, we go from 2N to N. We go from diploid to haploid. We get half the number of chromosomes. There's only 23 in a sperm. There's only 23 in an egg. In mitosis, we stay diploid to diploid. And the reason why that happens is because we make the copy ahead of time. We make the sister chromatids so that each cell ends up with 46 at the end. We start with 46, we end up with 46. Here, we start with 46, and we end up with 23 in meiosis, because eggs and sperm are haploid. They only have half the number of chromosomes. Everybody good to go? Sex cells, man. Oh, that's sexist. Could be woman. Anyway, let's see what we got here. Cell cycle, interphase. Yeah, okay, interphase, whatever. G1, S, G2, we'll talk about those. Yeah, let's take a look at the cell cycle. So normally, like freshman year when I drew the cell cycle, I drew it with mitosis on top. Campbell likes to put mitosis on the bottom. So we're just gonna do it Campbell's way, because some of the images I'm gonna show you later with regulation show it on the bottom, and otherwise it's gonna get confusing. So we're gonna put S on the top now, and we're gonna put mitosis on the bottom. And that's backwards from freshman year. We're still going clockwise. So this would be G1 here. This would be G2. Okay, so you just got to get used to it that it's upside down from freshman year, but it's a little confusing at first, but it's not so bad. Not so bad, not so bad. If you remember, perchance, actually, I think I made myself a blank slide here. Yes, I did. Okay, I'll get to that in a second. During G1, it's mostly growth. So mitosis happens here in M. The cells split. So we have a large cell going into two small cells. Those two small cells have to grow, okay? They have to grow into a big cell again, and that's what happens here. So more organelles form, more cytoplasm, membranes. The entire cell has to, expand, all of that stuff. S is DNA duplication, so we call this replication. And then G2, we prepare for mitosis. We have maybe a little bit more growth, and then, so mitotic machinery gets prepared. Mitotic machinery gets prepared, spindle fibers, microtubules, centrioles, all that stuff gets ready during G2. And G stands for gap. I don't know why it stands for gap. That doesn't really make sense, but that's just what it stands for. S is synthesis, M is mitosis. Okay, the one we wanna think about or add on here is G0. So that'd be up here then, G0, or GO, some people say. G0 is a normal cell. Like that's what a normal cell is doing. My muscle cells, my brain cells, most of the cells in my body are in G0. They're just doing their job. My pancreas cells now are cranking out insulin. They're doing, this and that. They're making glucagon. They're making digestive enzymes. They're in G0. They're not doing mitosis. Sometimes kids think the cell cycle is what cells do all the time. It's like, oh yeah, cells are always doing the cell cycle. They're always, it's like a clock and blah, blah, blah. It never stops. Wrong. It's most of the time they're not doing that. This is very special. That's only when mitosis is necessary. So clearly when, from conception, those cells, yes, they're doing mitosis continuously because you're growing super fast at that point. But once you're, in adult stage, once you're fully grown, mitosis kind of stops in a lot of tissues in the body, and G0 interphase is where the cells are at. They're now doing their job. Because during mitosis, they can't do their job. If a cell is doing mitosis, all the DNA is coiled up and all this stuff, and it can't actually, synthesize proteins. It can't read the DNA. It can't do mRNA. Any of that stuff is not possible at this point. Cells are kind of useless during mitosis, but they are, focusing on reproducing. So they got that job to do, and then when they're done, they go back to G0, then they can do their job again. All right, so does like skin and hair cells do this continuously? Sort of. Some of the skin cells are dividing, some of them are not. But yeah, skin keeps getting replaced and the old dead cells kind of accumulate on top. So the outer layer of skin is dead, excuse me, it's dead cells that have accumulated because of mitosis. Hair follicle cells, so the hair itself is dead. It's not like doing anything. It's not, not doing mitosis obviously, but in hair follicles, the hair is fairly slowly, fairly continuously doing mitosis. There's cells in there doing that, causing the hair to grow. For babies, is mitosis always happening? In a lot of the cells, yes, but not in all the cells. Okay, but in a lot of tissues, when growth is happening, mitosis is going on a lot more than in adults. Okay, cell cycle. So here, if you're going to draw it from scratch, you just do this. It's pretty easy. So drawing it is one thing, but you should also know what's happening at each stage then, okay? So G1, G2, so mitosis is here. So this would be like your, prophase, metaphase, anaphase, telophase, all that stuff that's happening here. And then cytokinesis. And then G1 is that growth, S is synthesis, replication, G2, your G0 would be here. Okay, so that's something you ought to be able to draw and explain what happens at each stage, please. Any questions? And I guess it would be nice if you saw the big picture, like if you realize this isn't normal for cells, like cells aren't always doing this. Most cells are not doing this. That'd be kind of a nice thing. Because I think a lot of times kids like go to biology class and they learn these things and then they memorize stuff like this and they don't really get the big picture and they get a misconception. And maybe that's my fault. Like maybe I don't emphasize that enough that, I don't know. I honestly don't know. All right, stages of mitosis. This is old stuff. The only thing is like Campbell breaks down prophase into like they add an extra phase, pro-metaphase. I put parentheses around it because it's like, who cares? Like, come on, stop. The College Board does not care about these stages, like you knowing what's the difference between prophase and pro-metaphase. You do not need to know that for the test. Don't worry about it. So the Campbell thing, I'm kind of going to ignore. I'm just going to put all the prophase stuff here. All right. Metaphase is when it lines up in the middle, when all the chromosomes are lined up. Anaphase is when they're splitting. And then telophase is when the entire cell is going to split, finished with cytokinesis. All right, and I got all the answers here for you. So everything that happens is listed here. So you don't actually even have to write it down. It's already in your PowerPoint. This PowerPoint is on Google Classroom if you haven't noticed that already. It's there for you. So 5 major things during prophase. Chromosomes condense, so that means they coil up into the into this, and it would actually be better to draw them like this, real close together, because the cohesins still have they're still connecting them. Centrosomes start movement, so centrosomes are the centrioles, microtubules, which you learned about in chapter 6. So the centrioles and some spindle fibers start to form, and it starts real small, and then these expand and they venture towards the opposite poles of the cell, to opposite ends of the cell, because they're going to be responsible for pulling apart the sister chromatids. All right, so the centrosomes form, start their movement. The nucleolus breaks down, because the nucleolus it can't stay. It has to break down, and then it reforms into 2 new ones, which is kind of awesome how it can do that. These things can just sort of fall apart and then reassemble. Like, wow, how does it do that? It's crazy. It's kind of like Star Trek. It's kind of like Star Trek, because I don't know if you ever watched Star Trek, but when they go on a planet, they don't like jump on a parachute or like they don't land and whatever. They just beam down. They just beam down. They like particleize the body into tiny little like whatever, and then they send it down as a wave, and then the body reassembles with the brain and everything intact on the planet, and then they're human again or whatever, and they can do stuff. And so it's pretty awesome. So that's what these things do. That's what your organelles do. Like they break down and reassemble. It's like, what? That's amazing. Is nobody impressed by that? You guys don't look impressed. Star Trek doesn't impress you? Okay. Yeah. I'm a huge Star Trek fan. I'm like a big Star Trek geek. Darth Vader is my favorite. I just love it. Anyway, yeah, nobody got that joke. Okay, yeah, that was lost. Okay, nucleolus breaks down, spindle fibers start to form. Oh, you know what? I'm missing one. The nuclear envelopes breaks down, but that's over here in pro-metaphase. So the nuclear envelope breaks down. That's like your 5th thing that happens. All right. Let's actually look at the pictures because it's better. So here we still have interphase, prophase here. The chromosomes start to show up. Normally they're all spread out like this. When they're all spread out, you can't really see them. It just looks like this shady, like a dark material that's chromatin. It's all spread. Then over here, they start assembling and you can start to see the sausages forming. That's these X's then. And they're all close together because of the cohesins here. So you start to see them. All right, the centrosome forms, which is the like spindle fibers, aster fibers, and the centrioles. And they start to move apart. Okay, so that happens. Nuclear envelope starts to break down. That's during pro-metaphase or at the end of prophase, let's just say. All right, eventually these reach the opposite ends and they're moving around these pairs of chromatids here, these sister chromatids. The chromosomes are getting moved into position because they have to all line up in the center of the cell. So all of that stuff happens during prophase. We'll just say this is all prophase. It's just a lot easier. Because why do we need an extra stage? Give me a break. Come on. Metaphase, they're all lined up in the middle. And if this is mitosis, there ought to be an even number. If this is a regular sexually reproducing eukaryote, there should be an even number. 1, 2, 3, 4, 5, 6. Why does it have to be an even number? 6, 8, 46, whatever. Because if it's sexually reproducing, half of them came from
Mom,
and the other half came from
Dad.
And that means if 2 contributed an equal number, even if those are odd numbers like 23, if you get 23 plus 23, it has to be an even number. That's just math, boys and girls, okay? What happens at the end of metaphase is the cohesins are getting cleaved. They're getting split and like destroyed. And then in anaphase, the cohesins are all destroyed and then they get pulled apart. Some people say in the beginning anaphase, the cohesins get split. I don't really care where you attribute that, but the cohesins have to go away. And then the kinetochore fibers that will, okay, so kinetochore fibers bind the chromosomes at the centromere. We call that region the kinetochore, all right? And so those fibers will pull these chromosomes in. Then there's some fibers that are called polar fibers. That's like these guys here. They don't touch a chromosome at all. They go from one end to the other. So those are polar. And the polar fibers push the cell apart to help split it later. These are kinetochore. Those pull. And then there's aster fibers. And the aster fibers anchor the centrosome to the membrane so that this is all stable and in place and can do the hard job of pulling those sister chromatids apart. So there's 3 kinds of spindle fibers, 3 kinds of microtubules there that take care of this. Surely you remember all your microtubules from chapter 6 that you studied and watched my videos. Anaphase is easy. They just get pulled apart and they start to separate towards the poles. And then telophase, the nucleus reforms again. So everything that happens in prophase happens backwards in telophase. So telophase is super easy to remember. All the prophase stuff happens backwards. And one extra thing happens is the cleavage starts. So the cell starts to pinch like this because what is pulling the cell membrane in? What part of the cytoskeleton? Is it intermediate filaments? Is it microfilaments? Or is it microtubules? What pulls the cell membrane together? Yeah, micro what? Which micro? Tubules or filaments? Microfilaments is correct. Nice job, Sophia. Very, very good. Okay, so it's microfilaments. But everybody knew that. You were just too shy to type it or to say it. Right, Sam Buckner? You knew that. Sam Buckner, you want to nod right now. If you want to live, you want to nod. Yeah, otherwise I got to push the button over here. Where's that apoptosis button? Nucleolus comes back, everything backwards, like I told you. Nucleolus comes back. Chromosomes disappear again. They unravel, they uncoil, opposite of condensing. The spindle fibers and the centrosomes disappear. They fall apart. Because all this stuff, like I said, it can fall apart and reassemble again. It's very, very cool how this can happen. Very, very cool stuff. Here I put a blank slide because I was going to draw all this for you. Yeah, I'm not going to draw this for you. You practice drawing it on your own. Where are we at? Where are we at for time? Oh, I got to go fast now. I only have 12 minutes. Oh, I don't know why. What time does this end? 1? We got that already. Aster, we already talked about. Metaphase plate. Okay, we did this. Oh yeah, here's just all the fibers that I already talked about. Aster fibers, kinetochore. Oh, I even spelled it right. Nice. Does it say polar fibers? No. Oh, it says non-kinetochore fibers. So the non-kinetochore fibers are the polar fibers. That's the same thing. That's these guys here. Okay, so they just have a different name for them, but that's the same thing. All right, anaphase we talked about, Oh, this is new information, Ensign. So this is something I did not teach you before. There were some scientists that wanted to know how exactly do the microtubules pull the chromatids? And there's 2 options. One, they're pulling them from the centriole, like pulling them in. And the other option is that they're shortening from this end, that they're shortening from here. And they just keep getting shorter, shorter, shorter. So what they did is they used a laser to mark them. They like darkened the area right here. And if they were going to shorten, this darkened area should move this way. But that's not what happened. So the darkened area stayed in the same place. And the microtubules just got shorter, 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 which means that the kinetochore actually has like a little Pac-Man mouth in it that chews up the microtubule. It destroys the microtubule a little bit at a time. These are almost like teeth. And it breaks it down and it gets shorter, 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 shorter. In the meanwhile, pulling this chromatid to the left. Okay, so the microtubule, like I said, can break down. That's no problem for it. Tubulin doesn't mind. It just hangs around in the cytoplasm and then it can reassemble again when it needs to. So you don't have to be sad for the microtubule. It's okay. It doesn't mind. It's kind of like chickens when they get made into chicken nuggets. They're happy to do that. It's an honor for the chicken. They're excited. All right. Good analogy, right? That was pretty good. All right. Cleavage furrow. Yeah. Okay, so this is when those microfilaments are pulling in the membrane and causing cytokinesis. So at the very end, when the cell splits, we say that's cytokinesis. And that's thanks to microfilaments. Plant cells do it a little bit differently because they can't pinch because the cell wall stops them from pinching. It's too strong and it's too rigid. So what happens is cell wall material forms in the middle. Cell wall material gets deposited in the middle, forms this cell plate, and then eventually that gets connected to the cell wall and it forms 2 half-size plant cells, which then have to, expand again and get bigger again. And plant cells have mechanisms of expanding that cell wall. They can make the outside of the cell a lower pH, make it more acidic, which loosens the cell wall and allows it to expand, makes it kind of more flexible. It expands, and then more cell wall material gets added, and the plant cell grows. It's pretty clever how they do that. All right, freshman year, we looked at onion root tip slides. Not really going to do that this year. I suppose I could make you do this on your own, but what's nice about those is like the stages of mitosis look exactly the way we expect. Like they look like pictures out of a textbook. It's so great. Like prophase looks exactly the way prophase should look, with the nuclear envelope breaking down, the chromosomes coiling, nucleolus is breaking down, all that stuff. Metaphase looks just like metaphase. They're all right in the middle. Anaphase looks like anaphase, where they're all getting pulled apart. Telophase, you can start seeing the new cell plate forming and the cell splitting, and the nucleus is reforming over here. It's pretty sweet the way it works. It's exactly what we expect. All right, cool. Sim, shut up. Yeah, I agree with that. That's pretty good. Here's some more onion root tips. You can study those, and you should be able to recognize these. They're pretty obvious. I mean, if you see a cell like this with everything spread out like this, and like the nucleoli, there's several nucleoli here, that's clearly interphase. This clearly is prophase because the chromosomes are starting to be visible. The nucleus is expanding and starting to break down. So interphase is easy to see. These are like normal interphase. This is prophase. I don't know if there's another. Here's another prophase, anaphase, metaphase. Yeah. Super nice. Prokaryotes. Oh yeah, they just do binary fission. We don't call it mitosis because they don't have a nucleus. They do not have a nucleus, so it's not mitosis. It's binary fission. what they do is their chromosome is a loop, so it's that circle of DNA. So basically what happens is replication happens at one place that's called the origin, where a bubble forms, and then 2 copies of the loop are made through DNA polymerase, and eventually 2 circles like fall apart, they separate, they fall away from each other, and then you have 2 new loops of DNA. That's how it happens in bacteria, and then the cell can split. So it's way more primitive than with eukaryotes. It's way simpler, a lot less steps and a lot less stuff happening. Not as interesting. We got a couple of minutes. So I want to talk a little bit about like primitive organisms that are sort of in between bacteria and like animals and plants, that are real simple organisms that don't do mitosis exactly the way we do. And by studying those organisms, like yeast, like dinoflagellates, like algae and things like that, single-celled creatures, we can learn about the evolution of mitosis. So there's some protista that do mitosis, or it's not really mitosis, it's like ancient mitosis more. So bacteria are at the top. Let's look at dinoflagellates. That's like a single-celled algae, a real primitive organism. They don't destroy the nucleus during mitosis. They keep the nucleus. And the microtubules like punch a hole right through the nucleus, and they push to the outside of the cell and expand it. They actually expand the cell like this until the nucleus gets separated. And the chromatids are separated by attaching to the wall of the nucleus, to the membrane, to the nuclear envelope, and that's how they get pulled apart. So there isn't any spindle fibers pulling the sister chromatids apart. They just attach to the nucleus, and the nucleus gets pulled apart, and it pulls the chromatids apart like that. So that's not technically mitosis the way we know it, but it's also not exactly the way bacteria do it. It's kind of an in-between. So we think, mitosis evolved through some of these primitive mechanisms like this into the complex process that it is today. So that's really bizarre. Nucleus does not disappear in dinoflagellates. It's weird. And like these holes through the nucleus for the microtubules, super weird. Yeasts and diatoms. Diatoms is another kind of algae. Yeast are a single-celled fungus. Well, you know all about yeast with schmooze and all. They also keep the nucleus. They don't destroy the nucleus, but spindle fibers do attach to the chromatids. So that's a little bit more like our mitosis, a little bit more mitosis-like. And then a bunch of spindle fibers push the nucleus apart and then also push the cell apart and eventually cause it to split. Cause this division, where cytokinesis happens and the cell splits all the way through. But the nucleus never really goes away. It just stays there and then just also pinches into 2 nuclei, 2 smaller ones. So that's diatoms and yeasts. And then finally, most eukaryotes. So these are eukaryotes. These have a nucleus, obviously. But most eukaryotes, just about all of them, do mitosis the way we know it. Normal mitosis, where the nucleus disappears, the chromosomes line up in the middle of the cell, and they get split by microtubules, all of that stuff. All right. Okay, we actually got done with the first part, and this starts the second part. The only problem is this is regulation and then a whole bunch of talk about cancer. So I'm going to need the entire time on Tuesday to go through this. I'm going to have to rush a little bit, but I will stop now. Let's end the share here and let's see if there's any questions, comments, concerns, criticisms, outbursts of hatred towards me, anything like that. No one? Okay, so what am I expecting you to do over the weekend? I'd like you to study. I'd like you to start studying. I'd like you to do the reading guide. I'd like you to start working on those free response questions. Maybe don't do them all, but do a good chunk of them. Do the ones that you already know how to do. What else? Study what? I don't know, maybe chapter 6, 7, 11, 12. Do we memorize all those types of mitosis? Just know that primitive creatures like yeasts and some algae don't destroy the nucleus. They do it differently. You don't have to be able to draw those or, explain them fully, but just know that it's in between what we do now and what bacteria do. Okay, so you don't have to memorize all those weird kinds of mitosis, but know that the nucleus stays intact in some of them. So I could ask you a multiple choice. I could say in dinoflagellates, this happens, and then, the options are the nucleus breaks down or it doesn't, and then you'd be able to figure it out. But I wouldn't give you a free response on that. Okay. Anything else? All right, I don't know what TSS is going to be like today. I think I might have a bunch of freshmen coming. I really never know what's going on, but if anybody needs to talk to me, you can always come to that. Just know that it might take a little while to get to you, because if a lot of kids want me at the same time, and it's a curse that I have, is everybody wants me. It's just being that popular. That's the problem. All right, if there's nothing else, you guys can take off and get started on your work. Crank out some of those free response. Have a weekend and see you Tuesday.